Okay, so we're going to be learning about how your cells make proteins. This is going to be split up into two lectures. Okay, this first one's going to focus on transcription, and the next one is going to focus on translation, which are like the two halves of protein synthesis. Okay, so let's talk about it. If you want to make a protein, your cell needs to receive some kind of message, right, through signal transduction, some kind of message, some way, somehow. The cell's going to say, oh, okay, I need to make this thing. And so we're going to need our DNA to do that, okay? But the DNA has the instructions to make the protein, but the DNA itself doesn't actually make the protein, okay? And proteins are made by what organelle? Ribosomes, okay? And ribosomes are in the cytoplasm, and DNA is in the nucleus. And we don't want our DNA to leave the nucleus, so we're going to need some kind of go-between. So we're going to give the instructions from the nucleus to the cytoplasm, and that's going to be RNA. Okay, RNA is going to take that message and those instructions and bring it to the ribosomes so that we can make the proteins that the DNA in the nucleus codes for. Okay, so the overall process here is you take DNA, you transcript it to RNA, and then it gets translated into a protein. So today we're going to be talking about transcription, how DNA comes into RNA, and then the next lecture will be translation, how RNA becomes a protein. Okay, so quick little review on RNA, right? Typically single-stranded. You've got a different sugar in there. You've got ribose, and you've got uracil instead of thymine. Okay. Again, we can still do complementary base pairing, still genetic material. It can still match up with DNA, which is exactly what it's going to do. So we've got three different types of RNA that we're going to be using in protein synthesis. You've got mRNA, which is called messenger RNA. This is going to be a complementary copy of a gene in the nucleus. You've got rRNA, which makes up ribosomes. Okay, so this is going to be what this right here, this is our rRNA. And then you have tRNA. This is transfer RNA, and so this is going to bring amino acids to the rRNA. So, okay, here we go. Transcription in detail. This is chapter 14.2 in your book, if you find this helpful. Okay, so again, transcription is making mRNA, that messenger RNA, from DNA. Okay, so basically we're going to have a gene that's turned on because the protein is needed. Okay, and so transcription is going to start. It's going to have three steps: initiation, elongation, and termination. Okay. So the very first step is called initiation. So we're going to look here in prokaryotes, right? Remembering that a prokaryote, very, very simple. We just got our circular chromosome in the middle. We got some cytoplasm and some ribosomes around it. Okay. So what's going to happen? Again, we're going to make RNA here. Okay. So the thing that's going to start off is RNA polymerase. Like when we were making a copy of DNA, we had DNA polymerase. Well, we're making RNA. So we're going to have RNA polymerase. And so this RNA polymerase is an enzyme, and it's going to recognize and bind to the promoter. What's the promoter? Well, here, okay, is your DNA, okay, your double-stranded DNA from your prokaryote. And the promoter is just a region, like right on the gene, or right in front of the gene. And this is like kind of like the start area. Okay, and so your RNA polymerase is going to find this promoter and is going to attach itself to the promoter. And then what's going to happen is your DNA is going to unwind, okay, little bubble. This should look a little bit similar, right? It's going to unwind, and RNA polymerase is going to, again, read DNA from 3' to 5', okay, and it's going to synthesize a complementary RNA strand. So here it is at the bottom, okay, it's being unwound, okay, and here is our new RNA strand that's being built. Okay. Now, one thing to notice is because RNA is typically single-stranded, we're just going to make this new strand. We don't need to do anything with this top strand, so there's no leading strand and lagging strand stuff, not like DNA synthesis, okay? So we're just going to use one strand, essentially the leading strand, okay? And we're just going to do it all in one big, huge chunk, which makes that a lot easier, okay? And then it's going to keep going. It's going to keep making this is going to keep going and keep growing until it hits the terminator. That's right. It's what it's actually called. Okay. So if here's the promoter where RNA polymerase starts, it's going to go and it's going to code till it hits this very special part at the end of the gene called the terminator. And this basically tells RNA polymerase, hey, it's about time to wrap up now. And when it gets to the end of the terminator, then RNA polymerase boop, pops off and stops making that new molecule of RNA. Okay, so those are the 3 steps in prokaryotes. Okay, so let's quickly look at it. RNA polymerase hops on the promoter, then it unwinds and starts reading it and builds your RNA strand, hits the terminator, pops off. Well, in eukaryotes, this is going to be a little bit more complicated. And part of this reason has to do with the fact that eukaryotes are just more complicated in general, right? Our DNA is going to be inside of a nucleus instead of floating in the cytoplasm, and this is going to become important later. Our DNA also isn't just one circular strand, it's lots of linear chromosomes. So to initiate transcription in eukaryotes, instead of having a promoter, this promoter is called a Tata box. I know, it's kind of crazy. This Tata box is right here. It's a whole bunch of thymines and adenines, T-A-T-A, in a whole row, okay? And so what's going to happen is a whole bunch of proteins called transcription factors are going to bind and kind of fold over right near this Tata box. It's going to make this bend. And then this bend is going to put it in the perfect shape that's going to allow RNA polymerase to come in and attach at the Tata box and start. Okay, so obviously a little bit more complicated here. So this is just another picture of this, okay? So your promoter in eukaryotes has this TATA, this Tata box, a whole bunch of transcription factors, which are just proteins, are going to come and bend and attach to these things called activators, which are earlier up. Basically, these activators are going to flip over right here. Okay, they're going to flip over so that everything is all kind of aligned right here. They form this perfect little pocket so that RNA polymerase comes in and is nice and cozy and can attach and fit on perfectly right here at the promoter at the side of the Tata box. And then elongation, it's going to unwind this whole RNA polymerase with some other proteins are going to unwind it. It's going to read from 3 to 5, build from 5 to 3, and it's going to make a single strand of RNA. This picture should look familiar because it's the exact same as the last one. So thankfully, elongation is the same in eukaryotes as it is in prokaryotes. We're just going to make one strand. There's no lagging strand. You just make one, and you're done. All right, so let's talk about how we terminate this process in eukaryotes. Remember, in prokaryotes here, we had the terminator where the RNA polymerase hopped off. In eukaryotes, it's a little bit similar here. Basically, your RNA polymerase is going to keep going till it hits this poly A signal sequence, okay? Basically, there's going to be a series of mostly A's right in a row, okay? And that tells your RNA polymerase, hey, it's about time to hop off now. And so after this huge long series of A's, some of the proteins that go along with the RNA polymerase are going to cut everything off, and your RNA polymerase is going to hop off, and now you're done. Okay, this piece of RNA now is not fully finished though. It's called pre-mRNA because it's not quite finished yet. Okay, with eukaryotes, we've got some what we call processing before it's considered to be totally done. So here's what happens. At the very, very end of your new piece of RNA, there's a whole bunch of like 50 to 250 adenines, just a whole bunch of adenines added to the end. It doesn't code for anything, okay? But what it does is it's going to protect. We're going to learn that there's going to be some enzymes that are going to want to break down this mRNA. And so this poly A tail right here keeps the enzymes from breaking down important bits in here. Okay, there's also going to be this cap ended on the 5' end. And that is going to help again, protect from the enzymes. And it's also going to help, because remember, this is in the nucleus and it has to get to the ribosome. So they're going to have to get out through the nuclear pore. And so this 5' cap is going to help it fit out the end. Okay, it's going to help it get through that pore. Okay, so that's going to be happening. And then also, we're going to have introns cut out. So in each gene, you have stuff that codes for something and stuff that codes for nothing, okay? The coding parts are called exons because they're expressed, and the non-coding parts are called introns. So the introns get cut out by these things called spliceosomes. So here's an exon, and you kind of get this little loop, and the spliceosome comes and cuts it out and then joins the two exons together, okay? So this might be what it looks like after it's made, but then once those introns are cut out, then this is what it looks like. So in this picture, the greens are your exons, the part that you want, and the reds are your introns, the part that you don't want. Okay, so that's going to happen. With prokaryotes, we have no processing needed at all because they're already in the cytoplasm. So you don't have to do anything special to get them there. They're already there. And so prokaryotes can already jump into translation immediately. In fact, sometimes simultaneously as they're being transcribed, which is very, very different because with eukaryotes, we got to get that mRNA from the nucleus to the ribosomes, okay? I'm going to stop right there, and then the next lecture is going to pick up and talk about, okay, now we've got a piece of RNA, like what do we do with it?